Hello everyone, I'm Fernando. I'm really happy to be here. I'm really to be hearing all of these different approaches that you have for mitochondrial transplantation. So I'm going to talk to you about the ischemia-reperfusion injury that we've been hearing a lot, but now let's just focus on the retina. Why the retina? Why do we feel that mitochondria have a critical role in the retina specifically? So one, they have a high energy consumption. They have the same amount of energy consumption comparable to the heart or the brain. They are really sensitive to a low oxygen deprivation time, and then these low ischemia times can lead to severe affections into the retinal functions. So what is exactly ischemia-reperfusion injury in the eye? This can be caused by a lot of things. It can be diabetes, obesity, dyslipidemias, age, hypertension, but basically what this means is we're going to get an occlusion at any point of the ischemia at the arteries that go through the retina, and then once we have this, we're going to have all of this cascade of events that are going to trigger inflammation, oxidative stress, VEGF secretion, and neovascularization as an effect of this VEGF. So the treatments that we have now is anti-VEGF, corticosteroids to try to diminish all of this inflammation, and some cases have hemorrhages, so we do a surgery to replace the vitreous humor. This happens to around 500,000 patients a year, and then just less than 5% of them recover visual function after treatment. And we believe that that's because we're focusing on downstream of the effect of the ischemia and the mitochondrial dysfunction, especially in the retina. So we are trying to focus a little bit upstream. For this, we decided this experimental approach where we're going to use the ARP19 cell line, which is a retinal progenitor human cell. We're going to try the uptake, functional integration, and different dosages of mitochondria. Then we expose for H2O2 to resemble the ischemic environment in the retina for either 1 or 4 hours to study different time points and different RNA transductions. And then we did ATP, live and dead assay, and RNA sequencing either after vehicle or mitochondria treatment. So this is the first result that we got after improving mitochondrial metabolism using nicotinamide in cell culture. And we could see that we can get in 15 days ARP19 cells that resemble primary cell lines, as you can see in the lower image. Then we did different dosages. We went to 1 times 10 to the 4 per every 10,000 cells to 1 times 10 to the 8. And as you can see, we had kind of a dose response, and we had significant ATP production at 10 times 10 to the 6 per every 10,000 cells. But we still wanted to confirm if these cells, if these mitochondria were actually inside. So we did the fluorescent imaging, and as you can see, they're actually collocating in the same network around the nucleus, and they do resemble different entities. But we still wanted to make sure that we were not seeing the membrane. So we wanted to make sure that we have the mitochondria inside of the cell. So as you can see in the lower, we have the C-stack representation of the 3D modeling of the cell, and we can see that yes, both mitochondria, either post or transplanted, are both inside of the cell, which is really encouraging. So moving forward, this is the dose that we're going to use for all of our experiments, 1 times 10 to the 6 per every 10,000 cells. So then let's go to the ischemic model. As I said, we chose 1 or 4 hours of stress at different concentrations. So we went to the lowest one, which is 0.1 millimolar of H2O2, and we went to the highest one, which is 1 millimolar. And then we could see here that we had an ATP depletion dose dependent for either 1 or 4 hours, so the effect was the same. And then after those 1 or 4 hours, we took the stress out, we washed the cells, and then we did the mitochondrial transfer at the dose that I mentioned. And as you can see at both time points, either 1 or 4 hours, we had some residue of ATP, depending also on the damage that was done. I really want to highlight that at high oxidative stress concentrations, we didn't see any recovery of the ATP either with vehicle or with mitochondrial transplantation, which really guides us into a threshold. Like mitochondria can only do so much into the different contexts that we have, as you can see in different diseases in the human body and different cell lines. So this just narrows a little bit of this application. So specifically, I want to highlight this specifically in retinal cells. So then we did the live and dead assay to see if this also correlated with the percentages of apoptotic cells. And we saw that yes, correlating with that, at higher concentrations we saw no differences, but in concentrations that we saw the ATP recovery, we also see reduction of apoptotic percentage of cells. So then we did the RNA sequencing, and as you can see, this is at the 1 hour time point. This is just gene ontology after RNA extraction. You can see we have upregulation of nuclear division, chromosome segregation, nucleosome assembly, and organelle fission, which was really interesting to see in our data. So this is guiding us that the cells at the 1 hour time point, we are getting some type of cell recovery, cell replication. So then we took these genes and we see that we have shifting of the genes sets from mitochondria. They resemble more the phenotype of a control cell rather than when compared to vehicle. So then we took these gene sets and we did some pathway analysis. And this is where I want to highlight. We saw downregulation of apoptosis, IL-17 signaling, IL-6 receptors, IL-6 signaling. So this is really encouraging because we're not seeing just ATP, we're not just seeing like complexes between, like inside of mitochondria, we're seeing an effect in all of the different systems within the cell and cell functions. This is at the 4 hours. We can see now that we're shifting away more to a pro-inflammatory, but not inflammatory, like an inflammatory response that's guiding us more into repair rather than modulation. We can see some reduction as well of apoptosis. We see stress adaptation. We see increased production of antioxidant, for example, glutathione. And then we see mitochondrial membrane regulation pathways also being activated. We then again took these pathways, these gene sets, compared them, and we can see that mitochondria treated ones were resembling more the control phenotype when compared also with vehicle. And in one group we saw that the mitochondrial transplanted one had a unique set of genes that were just modified in this situation. So we took this again and we saw that we had, again, the IL-17, we have downregulation of the MAP kinase signaling pathways, we have reduction of lipid metabolism, TNF signaling pathways, and the mTOR signaling pathway, which was also really interesting. We saw the DDIT4 being downregulated, and we see upregulation of SGK1, which we know that it's linked to cell survival. So just takeaways here of the genes that we saw and some of the pathways and the mechanistic insights that we observed were like the MAP kinase, the mTOR, the IL-6 and the IL-6 receptor. We saw downregulation of Cas9, COSB, and VEGF signaling, which is in this case specifically in the retina. We are really interested because we know that the treatment now is anti-VEGF. So if mitochondria we're giving it, it's acting both upstream and downstream of this effect. Of course, we know that every study has strengths and limitations. This study just covers in vitro modeling of the retinal cells, so this needs to go still to an in vivo setting and then hopefully to a human setting. We have some strengths that we developed this in a mitochondrial specifically focused lab, so we have the methods really well validated, and we have a lot of really exciting therapeutic potentials for the retina, which has been a long time forgotten for mitochondrial approaches. So in conclusion, we see that we have bioenergetic rescue, but we also see that we have a multi-dimensional systems intervention. We're not just recovering ATP, we're recovering ATP, but we're working in modulation of inflammation, modulation of the environment of the retina, at least as shown by our data. Few directions we are working now on getting this into an in vivo setting, and then we're also studying how we can get this faster to the patients. I don't want to go without saying thank you to both of my hospitals, Boston Children's Hospital, the Macaulay Lab, and Massa here at the Babas Lab. Thank you, everyone.
Excellent job. Does anybody have any questions?
Great. I have a question. Like Dr. Macaulay used direct intracardiac injection and saw results. It seemed like the eye would be an ideal organ to do direct ocular injection of some type of mito slurry. You could see some retinal effects pretty quickly with electroretinograms or whatever. Has anybody looked at anything like that?
Yeah, actually, we're working now in the modeling of the in vivo experiment approach, and actually the drugs that we have now for patients are all delivered intravitreally. So it's an injection that goes directly into the vitreous humor, and then it's dispersed all throughout the retina. So we would use the same approach as used in humans now. We will give the direct injection, and the size of the gauge allows us, it's the same size that we would use in a heart, so it allows the mitochondria to go by and remain intact after injection.
So, can I ask, so did you try this one on macular degeneration?
Yes, that's an interesting setting. For the purpose of this experiment, we just focus on the acute ischemia reperfusion injury in the retina, but it's really reassuring that once we get like some more results, this would also be translatable, because you know the in vitro models for AMD also focus on H2O2 concentrations for longer periods of time, and we did 1 and 4 hours, which is more into the acute setting. But yeah, it would be an interesting avenue to explore later.
Thank
Last
you.
question.
Yeah.
Up front here.
Thank you. That was a great presentation. So I was wondering, in your tissue model, do you observe a systematic uptake in every cell, or do you have like islets suggesting some sort of clonal favorable context? Or
Yeah, that's a really good question. When we go to the imaging, yes, we do like a full scan of the area that we are going to take the picture from, and then you can see that the uptake is really widespread. We do not see like just in some areas, so that's encouraging as well. Thank you, everyone.
Excellent.